Welcome, everyone. My name is Josh Summer. I'm the co-founder and executive director of the Chordoma Foundation. Thank you so much for joining for this impact update. We'll go ahead and get started. A couple quick housekeeping notes. This webinar is being recorded, and if you have any additional questions or want to come back to it, you'll be able to see it online afterwards on the Foundation's YouTube channel. This should be thought of as an interactive conversation. I'm going to go through some slides and provide a number of updates, but please, if you have questions, don't hesitate to ask them throughout the presentation using the Q&A button at the bottom of your screen. I won't see the Q&A, but my colleagues at the Foundation will be monitoring that, can respond to you. And at the very end of the session, we'll also have some time for additional Q&A. Finally, if there are any additional questions or any feedback that you have for us, please, we very much welcome it, and you can reach us at support@chordoma.org. So a quick outline of what we're going to talk about today. For those who might be new to the Chordoma community or maybe are less familiar with the Chordoma Foundation, I'm going to spend about the first seven or 10 minutes sharing a brief background about the Chordoma Foundation, what we do, and big picture progress that's been made over the last 14 years. I'm then going to focus much of the update on some very exciting recent progress, as well as upcoming progress and opportunities focused on the Chordoma Foundation's 5 strategic priorities, and really highlight advances that have been made possible by the support and investment of the Chordoma community. And then, as I mentioned, finally, at the end, we'll save some time for some Q&A. So for those who I don't know or who we haven't interacted, a bit about me. It's hard to believe, but I'm now a 15-year Chordoma survivor. My involvement in the Chordoma Foundation is very much born from a personal sense of urgency. I was diagnosed when I was 18 as an undergrad with a skull-based chordoma. And at the time, I never would have imagined being here 15 years later and getting to experience the joys of having a family. But I feel just exceedingly grateful to have beat the odds. And at the same time, continue to feel an even heightened sense of urgency, knowing now what is at stake, but also having unfortunately seen many fellow patients and members of the Chordoma community deal with the disease even after 15 years, 20 years and longer. And sadly, some of them even succumbed to the disease after that time. So continue to feel a very, very significant personal sense of urgency. The Chordoma Foundation got its start about 15 years ago and really grew out of the efforts of a small number of families that came together, which were united through an online support group at the time. And all of us shared a sense that the options that were available to us were not good enough, that the odds of recurrence were too high, and that if the tumor recurred, the options available were inadequate. As I'm sure many of you know, chordoma is primarily treated with surgery and radiation. Some patients are lucky enough to be cured, but for those who are not cured initially and for whom the tumor does recur, the treatment options are really limited. And not only are they limited, but oftentimes treatment can lead to significant impact on quality of life. The diagnostic and treatment odyssey to get the right care is oftentimes very challenging. There's a ton of challenges that come along the way. And so we realized that there was progress that was needed. We found the situation to be unacceptable. And that by coming together, we might have a chance of creating a better future for those of us who are affected by this disease and those who would come next. I want to just highlight for a moment one of these individuals, a dear friend, Michael Torrey, who was really a huge part of bringing the Chordoma Foundation to life. He was a fellow skull-based chordoma patient, one of the Foundation's most dedicated and generous supporters, just a huge optimist and a believer in the progress that was possible. Michael battled chordoma over the entire course of the Chordoma Foundation's existence, and sadly, actually just yesterday, passed away, not actually of chordoma, but of COVID, which is really, I think, a cruel twist. But I just feel tremendous gratitude to Michael for his encouragement, his constant sense of optimism, his generosity, and really all that he did to make the Chordoma Foundation what it is today. He was perhaps the biggest believer in what was possible. And I think it consistent with Michael's vision and really the vision of all of us who came together, we wanted for one day for everyone affected by this disease, not only to be able to overcome the disease, not only to be cured of their disease, but also importantly to maintain their quality of life. And that's what we've been dedicated to for essentially the last 15 years. Our mission to achieve that vision one day is to improve the lives of those affected by chordoma and lead the search for a cure. And delivering on that mission, what we've come to realize requires work in 3 vital areas, and not in isolation or separate, but really intimately integrated. Each of these areas is essential and mutually reinforcing. So that's research to find better treatments, healthcare improvement to promote better care, and patient services to enable patients to have the best experience possible throughout their journey with this disease. Because at the end of the day, even if we find better treatments, if doctors aren't providing them to patients, if they're not giving patients the best care possible, ultimately that's not going to lead to patient benefit. And furthermore, if patients can't access that care, if patients can't get the support that they need along the way, those better treatments are not going to live up to their full potential. So this is a comprehensive approach that really goes hand in hand. I want to just for a moment talk about how the Chordoma Foundation is different from traditional medical research foundations. As you may be aware, there is a very tried and true model of nonprofit disease-specific organizations whereby patients and family members come together and raise money, and the organization offers up grants and hopes that talented researchers apply for those grants. This is important. It serves an important need, but by itself is not sufficient. And the reason being is that there's a whole sequence of research that's needed to go from an understudied, poorly understood disease like chordoma ultimately to better treatments and a cure. You could think of that as research needed from A to Z. And oftentimes what happens is when you put out these requests for proposals, you'll get proposals for research to do A, B, and C, and J, K, and L, but there's huge gaps in between because the infrastructure, the critical enabling resources such as tumor tissue or cell lines or animal models don't exist. Or maybe the researchers who have the relevant expertise aren't yet aware of the disease or aren't interested or aren't motivated to study it. And so we take an end-to-end approach, ensuring that all of the relevant research at every step of that R&D development process gets done and is feasible. Additionally, especially for rare diseases, research tends to be slow and costly, and there's a lot of reasons for this. Research in general is driven by individual investigators who are not necessarily talking with one another. Sometimes they're duplicating effort. A lot of times research in individual labs is done at a small scale and is therefore not optimally efficient. And additionally, it takes a long time for data and materials to flow between labs. And so given that all research is cumulative, every discovery is based upon previous discoveries, those lags in sharing between labs can really cause the process of discovery and therapeutic development to become protracted. And so we at the foundation try to take a proactive approach to find ways to make research faster and more efficient. So what does the strategy look like? Ultimately, this is a strategy that is driven by the urgency that we feel as patients. It is not the typical academic research model that is oftentimes slow and plodding. It's essentially a two-pronged approach. Number one, create a conducive research ecosystem to make it possible for the most talented researchers and companies to work in this space, to bring their expertise and capabilities to bear on chordoma. And that means things like building a research network, developing repositories of tumor tissue and cell lines and animal models that researchers can easily access. It means developing a set of shared research services so that experiments that need to be done over and over and over again in multiple different labs can be done quickly and efficiently, rather than having each lab have to recreate those same capabilities. And then within that context of a conducive research ecosystem, ensuring that the most important high impact and high priority projects get done. We don't believe to be or claim to be aware of every single important project, but we do know how to listen, and we continually are polling and soliciting from the research community ideas about what the highest priority projects are, and then we work with our medical and scientific advisory board to distill those down and ensure that those projects are proactively addressed, not leaving it to chance for researchers to happen to take them on or happen to become interested. So with this approach, and having invested now about $15 million in research and leveraged more than 25 million from other funders, whether that be companies or the NIH or other foundations, the field has come a long way in the last 15 years. We've gone from having essentially none of the basic materials that researchers need to study the disease to now having abundant cell lines and animal models, a source of tumor tissue that researchers can access. Those resources have now been utilized by over 100 labs across the world. And in doing so has enabled just a tremendous number of discoveries and a much greater understanding of the biology of the disease. This has culminated in the discovery of more than 35 therapeutic concepts, over 60 drugs tested in mice. And we've gone from having one drug that was tested in clinical trials when the foundation was started to now having more than 10 clinical trials specifically for chordoma get underway, including 6 that the Chordoma Foundation has directly helped initiate and support. This is all fueled by a small but mighty community. We know that chordoma is a rare disease, but when we come together, we're seeing that really powerful things are possible. There are now more than 5,000 patients and family members who have come together in this network. There are more than 300 researchers now that are studying chordoma. The progress that we've talked about has all been fueled by more than 13,000 donors, as well as over 150 volunteer fundraisers, including, I'm sure, some of you who are joining today. This is all also made possible by quite a number of volunteer leaders, from our board of directors to our advisory boards, to our ambassadors and the moderators of our online community, et cetera. And then a tremendously dedicated and talented professional staff of 10, ranging from PhD scientists to social workers to communications professionals and everything in between. So I want to focus now on talking about progress that's been made in recent years against the Chordoma Foundation's 5 strategic priorities. These are priorities that we've been focusing on now since 2017, and they are intended to achieve impact, achieve patient benefit, both in the near term and in the long term. And they're arranged here essentially in order of their impact on differing time horizons. So in the near term, working to improve the patient experience through our patient services, et cetera. And over the long term, working to develop better treatments and potentially even curative treatments. So now I'm going to walk through one by one and talk about some of the exciting progress that this community has made possible in each of these areas. First, improving the patient experience. I think, as many of you are probably aware, over the last 14 years or so, the Chordoma Foundation has built up quite a suite of services for patients to help identify appropriate treatments, to help connect with experienced physicians, to help connect with other patients, et cetera. But much of what was available was focused on addressing the needs that occur when patients are either diagnosed or in need of treatment. But what I think has become abundantly clear, and probably many of you can attest to, is that even when you're not dealing with treatment in a kind of an acute sense, chordoma is still with you, whether it be the disease itself or whether it be the physical or emotional effects. The dealing with chordoma really doesn't end for most people after treatment ends. And importantly, we recognize an ongoing need for support, guidance, assistance of a variety of types. And now the Chordoma Foundation, through our survivorship initiative, is here at every step of the journey. There's a wealth of resources now available on our website. You can go to chordoma.org/survivorship to learn more, everything from educational resources to a directory of specialists who deal not only with the treatment of chordoma, but of the side effects and various quality of life issues that are associated with chordoma, and a number of other things as well. I'm really excited this year to have launched 2 new virtual support groups, both for patients and survivors, as well as for caregivers and co-survivors. And if anyone is interested and hasn't yet participated, very much encourage you to check it out. We have really, really strong moderators, professional social workers and therapists who are quite experienced at leading support groups. Additionally, as some of you may know, we typically put on community conferences, either international conferences or regional conferences, but this year, because of the pandemic, we moved those to a virtual conference series. We've just concluded our fifth webinar in the series. Essentially, all of these webinars have been focused on survivorship issues, everything from dealing with pain to fatigue, anxiety and depression, balance, etc. All of these are available on our YouTube channel and on the website, so definitely encourage anyone who's interested to check those out. I'm really excited also about an initiative that we've undertaken last year, and many of you may have participated in, to really try at a detailed level to understand the survivorship needs and challenges that patients and their caregivers face. So we've had an incredible over 850 participants take our survivorship survey. This has just provided a wealth of insight into all sorts of challenges, whether they be physical challenges, emotional challenges, social, etc. And importantly, we're also assessing the extent to which those challenges are being met. And essentially the take-home message here is that you can see here in these pie charts in orange, that's the proportion of people who report that indeed their challenges are being met. So clearly there's a lot of work to be done, and the results of this survey are already informing the development of additional programs and services that we're going to roll out in the coming year as well as beyond. And then I'm also really excited to share that these data are being analyzed in a really systematic way in partnership with the research group at Johns Hopkins, with the goal of publishing these findings so that it can serve as motivation for physicians and other care providers, perhaps to focus their efforts to address the survivorship needs of patients and caregivers a bit more as well. So moving on now from addressing needs in the very near term to identifying better ways of caring for the disease and better treatments for the disease. As I'm sure all of you can relate, there's a lot of questions about chordoma that still remain to be answered. We know that this disease, every patient's journey is unique, and at every stage of the disease, there's a wide variety of potential outcomes. There are patients who do really well, and there's some patients who sadly experiences fast recurrence and progression. We don't really have a handle yet on that full range of outcomes, and importantly, what might predict them. But these are questions that can be answered. We also need to understand urgently, are there any experimental therapies, off-label therapies, possibly even alternative medicines that patients are benefiting from? We certainly hear anecdotes about this happening, but the problem is anecdotes can't be generalized, and you really need a systematic analysis to be able to make sense of all this. I really want to underscore that because of how small the chordoma patient population is relative to other more common cancers, literally every single patient's experience is informative, and there is something to learn from everyone's experience. So I'm really thrilled that over the last couple of years, we've been able to partner with the National Cancer Institute, a very dedicated group of researchers there who are focused on studying rare cancers. And we've been able to get chordoma included in an ongoing natural history study of rare tumors. A natural history study essentially is an observational study, so it doesn't involve any treatment, but it involves following patients over time to observe how their disease progresses, how they do, what side effects they have, et cetera, what treatments they respond to. And importantly, this also includes a biological component whereby tumor samples will be collected to see if it's possible to correlate aspects of a patient's tumor with various outcomes that they might have. For example, length of survival, recurrence after treatment, or response after treatment, et cetera. So this is open to every single chordoma patient anywhere in the world. It's free to enroll. Once you enroll, it's a simple enrollment process. And once you enroll, the researchers at the National Cancer Institute essentially take it from there, interfacing with your healthcare provider. So it's a really light lift and something that every single one of us can do to help advance understanding of the disease and ultimately better treatments. In parallel with this natural history study, the Chordoma Foundation, thanks to the generosity of many in this community, we're supporting 5 ongoing projects, each of which are employing complementary approaches to try to unravel or decipher the biological differences between different chordomas. So for example, are there differences between tumors of different anatomic locations, skull base versus sacral, for example? Are there differences between pediatric and adult chordomas? Are there differences between tumors that metastasize and tumors that don't metastasize? And what might drive tumors to metastasize? All of these questions require creating a full picture of the biology of the disease, essentially a multi-layered picture. The way I think of it is like a multi-layered map. You've got your topo layer, you've got your roads layer, you've got your satellite layer. Each of those layers individually only paints part of the picture, and it's only by layering them all on top of each other that you really get the full picture. So that's what these groups are doing. They are applying their own kind of unique expertise and capabilities, but we're also connecting them with one another and coordinating these efforts because there is a lot of potential synergy between the work that they're doing. I want to highlight some really exciting findings that have come out about different biological subtypes of chordoma, which could be either in the near term or longer term clinically relevant for patients. So the first on the left, what you're seeing here is a study from the University of Pittsburgh where they found that there was some specific markers within chordoma tumors that seemed to be associated with better or worse outcome. And when they looked at these markers systematically in over 100 chordoma patients over a multi-year period, they found that indeed there was a very strong correlation between the presence of these markers and and outcome. So overall progression-free survival. Importantly, what they also found was that these risk groups were predictive of whether patients would benefit from radiation after surgery. So essentially the take-home message is if you fall into the high-risk group, group C or group or the intermediate group, group B, there is a clear benefit to getting radiation in terms of improving progression-free survival. However, if you're in group A, if you're in this low-risk group, getting radiation, at least in this cohort, and this needs to be confirmed in a larger cohort, but at least in this cohort did not make a difference. And so perhaps those patients could avoid the risks of radiation. What's really exciting is that there is, in fact, right now, a pathology test that's available at most large hospitals where patients who are newly diagnosed could actually have this test done. And this could potentially inform, how aggressive to be in the initial setting, in particular, whether radiation is warranted. I do want to emphasize, however, this is a single institution, and these results need to be validated at more institutions and in a larger cohort. It was also only skull-based patients. And so a big priority right now is to extend this study to include not just skull-based patients, but also patients who have tumors elsewhere. And there's 7 different hospitals who have come together and submitted a proposal to apply this same test retrospectively to over 1,000 banked chordoma tumor specimens. And that would answer definitively whether, in fact, this could help guide treatment. So that's a proposal that we're very much hoping to be able to support here in the coming months. Additionally, on the right side of the screen, there's a very exciting study that's come out of a group that we supported in partnership with the Canadian Cancer Society at the University of Toronto. And what they have found is that there are essentially 2 different biologic subtypes of chordoma. So not just looking at an individual or a couple markers within the tumor, but actually at a kind of a global level, it seems as though there are kind of, you could almost call them different flavors or different varieties of chordoma that are evident through what's called epigenetic signatures. So this is not necessarily changes in the DNA sequence, but changes rather in how the DNA is organized and how different genes are turned on and off. And importantly, these different groups, they are associated with different prognoses, but also because they have underlying biology, it's very possible that they might require different treatment approaches. Another exciting thing about this is that this group in Toronto found that these different subtypes could actually be detected through a test involving plasma, so through a blood sample. So you don't actually even need to have a biopsy or surgery to be able to determine A, to diagnose chordoma, and B, what subtype you fall into. I want to emphasize, though, that this is very, very early. This is still a research study. This test is not available clinically, but with continued research in the next several years, it very well could be available clinically. Okay, now I want to turn to a topic which is very timely and very near and dear to my heart, which is drug repurposing. And essentially, drug repurposing is about asking the question, are there any drugs that already exist that could benefit chordoma? We're certainly seeing, for example, in response to the COVID pandemic, drug repurposing starting to bear some fruit. There's some existing drugs that are showing some promise in clinical trials for treating COVID. And certainly the same could be true for chordoma. We don't know until we look. And because there are lots of existing drugs, more than 2,000 that are already approved, as well as like 4,000 more that have entered clinical trials, if you're just kind of cherry picking or looking at a few, you're likely to miss a lot. And so a systematic approach is definitely warranted. So we've worked with researchers at the Broad Institute of Harvard and MIT to screen what is one of the most comprehensive repurposing libraries anywhere in the world. Here you can see a breakdown of the different types of drugs that are included. So it includes about 2,000 drugs that are already approved, as well as others that are in clinical trials, et cetera. These include drugs for psychiatric disorders, for infectious disease, cardiology, the whole gamut. Really, we are completely agnostic as to where these drugs might come from and want to find anything that could be effective. So that effort has already yielded some interesting hypotheses, at least 3 promising therapeutic classes that seem to be showing activity in cells. Further research is needed to validate this, but at the end of the day, that is but one effort to identify existing drugs or existing drug targets. And at the end of the day, this is going to need to be tested in patients. The challenge is there's a limited patient population. So, only so many clinical trials are ever going to be able to be done, not only because of limited patients, but because there isn't unlimited funding. And moreover, if you're a patient, you want to make sure that the clinical trial that you're going on is not just a good one, but is is better than any alternatives. And so we think that the bar ought to be high for bringing therapeutic concepts from the lab into the clinic. So how do you assess which therapies to prioritize for clinical trials? And the answer is there's a lot that one can learn from testing drugs in cells, but the best model that we have right now to predict response in patients is what's called patient-derived xenograft models. These are human tumors that are taken from a patient and implanted and grown in mice. And that's really the linchpin. That's what drug companies look at. That's what most granting agencies look at. That's what most researchers want to see to move forward with a clinical trial. I should also mention most IRBs, these are ethics boards. You want to be able to have a pretty strong case based upon animal data that the drug is going to work in patients. The challenge is testing drugs in animal models is costly. It's generally slow and inefficient when it's done in academic settings. And to do this over and over and over again in various academic labs is a tremendous duplication of effort. So over the last 7 years, we built a program which we call a drug screening program that allows for the rapid, systematic, and cost-effective evaluation of drugs in these PDX models. And so far, we've received over 100 nominations from all researchers all over the world, researchers we've funded as well as other researchers, and in addition to that, companies. Out of those 105, 82 have been selected by our scientific advisors as being sufficiently promising and sufficiently well justified to take into testing. And here you can see we've started testing over 70. We've completed 49. 5 of those have advanced now to the point of being nominated for a clinical trial. 4 of those clinical trials are ongoing, and 1 of them is in the planning process. This pipeline is only continuing to grow. There's 23 concepts that are currently being tested, and I'm really excited. It's too soon, I think, to take this to the bank or to really celebrate, but there are 4 additional concepts that are coming down the pike, which are looking really quite promising in some of the initial models in which they've been tested. We need to do some confirmatory studies, test these in additional models. But if the data holds up, it's very likely that at least some of those concepts will be nominated in the next several months to the Medical and Scientific Advisory Board to be moved into a clinical trial. In addition, there's 10 concepts that haven't yet started testing, which are teed up. Some of these are ready to start soon. Others are pending additional funding. And then we're constantly accepting new nominations from researchers across the globe. I just want to give a couple examples of the output of the drug screening program and what this data actually looks like. What is this telling us? So here, what you're seeing is these are the results of efficacy studies done in mice. The black line is the growth of tumors in these mice over time. And as you can see, untreated tumors, they grow at a pretty steady rate. Most tumors grow, they grow exponentially. What you're seeing in blue is imatinib. So many of you may have heard of this drug. It's also called Gleevec. For many years, this has been the kind of standard go-to systemic therapy when tumors recur. We've known based on clinical trials that the activity of Gleevec is pretty modest. Maybe some tumors stabilize, but for the most part, you don't see tumors shrinking. So in red, what you're seeing here is tumors that have been treated with the drug afatinib. And there you can see very significant tumor growth inhibition, even perhaps a little bit of regression in one of these models. I think it's important to note, though, there is variability between models. Just because in one model it works doesn't mean it works in all models. And so it's really important to always take this preclinical data with a grain of salt. There's a truism in research that says all models are wrong and some are useful. And so at the end of the day, even if something works in models, it needs to be tested in clinical trials. But certainly, this provides quite strong rationale, and indeed has provided the rationale for an ongoing clinical trial with afatinib at 3 sites in Europe. Now the priority is finding drugs that that don't only cause the tumors to flatline or to stop growing, but actually cause the tumors to regress. And more than likely, based upon what we've seen across the field of oncology for other tumor types, more than likely this is going to require combinations. So a big focus right now is finding rational combinations that can work together to drive greater tumor growth inhibition or regressions. Here you can see on the right some very encouraging new data. This literally just came out about 2 weeks ago. And I just want to note, the reason I'm able to share this to you, even before it's published, is because the Chordoma Foundation, this is data that we own. And unlike academic researchers, our success is not measured by publications. Our success is measured by how quickly beneficial treatments reach patients. And so, we have an incentive to get this data out there as quickly as possible to researchers so that it can be acted upon and clinical trials can be started if warranted. So combination is very important now. Also identifying mechanisms of resistance to drugs that are in clinical trials already. We want to see if we can identify biomarkers that predict which patients are going to respond to which therapies. And then importantly, we're also right now developing new mouse models that are capable of being used for testing of immunotherapies, which are coming down the pike. That actually brings me to the next priority, which is that of immunotherapy. I think probably many have heard about the extraordinary impact that immunotherapy is making for subsets of patients with other tumor types. So in the last 5 to 10 years, there's really been a sea change where there are a handful of therapies that can make a dramatic impact, not for all patients with certain tumor types, but for those that benefit, the benefit tends to be really significant. We're seeing some promising initial hints that chordoma might be one of those tumor types that can benefit from therapies that harness the immune system. So to briefly summarize, there's just this year, there's been some really promising data. First, that came out of a phase 2 trial in France, showing that about 10% of patients treated with Keytruda, also called pembrolizumab, had their tumors shrink. Just last week, there was a group at MD Anderson that we work with very closely that presented at a conference showing that 4 out of 17 patients treated with various checkpoint inhibitors had their tumors shrink, and 11 more of them had stabilization. So again, this is just a case series, not a clinical trial. So you can only put so much stock in this, but nonetheless, a very promising signal and one that deserves further attention. There are 2 ongoing clinical trials testing checkpoint inhibitors. One that the foundation is supporting ongoing at Hopkins, and then another ongoing at UCLA. There's also some very promising initial data that's come out of a recent vaccine trial testing a vaccine targeting Brachiora. As some may be aware, this is now actually the 5th trial testing a Brachiora vaccine, and the earlier ones were somewhat underwhelming, and the data was somewhat equivocal. This is a next generation, a 3rd generation vaccine where it's thought that it could be designed in a way to stimulate a more robust immune response. But all that being said, a lot of important questions that still need to be answered. Why are only some patients benefiting and not others? And what can be done to enable more patients to benefit? So to answer those questions and others, we are now supporting and collaborating very closely with 3 different research centers, Northwestern, MD Anderson, NYU. 2 of these, the Northwestern and MD Anderson projects, have been co-funded with the Cancer Research Institute, which is the premier funder of immunotherapy research. We're now actually right now accepting proposals for the 3rd year of this grants program, and are very excited about the new concepts or the new ideas that are incoming. So finally, I want to turn now to a topic that many of you may have heard of. That is this gene and protein called brachyury. So many probably know that brachyury is the definitive diagnostic marker of chordoma. Almost all chordomas, with the exception of dedifferentiated chordomas, express brachyury. But over the last decade plus, there has been an accumulation of evidence to suggest that brachyury is not only chordoma's calling card, but also the key driver and chordoma's greatest vulnerability. So if you look here in the bottom left, what you're seeing is an experiment whereby every single gene in chordoma cells, you have every cell in your body has about 23,000 genes. This was an experiment where every single one of those genes was turned off one by one to ask the question, which gene is are chordoma cells most dependent on? And then that list was compared with a wide range of other cell types. And the take-home message, what you're seeing here is that far and away, by a huge outlier, brachyury is the most selectively essential gene in chordoma. So it's the thing that chordoma is most dependent on, and yet other cell types are not dependent on it. So because of that, brachyury is now oftentimes referred to as chordoma's Achilles heel. It is this unique and absolutely essential vulnerability. And so it's natural to think, okay, given how important brachyury is for chordoma, that, could this be a drug target? If you could somehow shut down brachyury, might it actually stop or kill chordoma cells? At the same time, there's been quite an emergence of evidence suggesting that brachyury is also important in quite a number of other tumor types. The role that it plays in these other tumor types is probably different. It leads to progression, resistance to therapy, metastasis, et cetera. Whereas in chordoma, it seems to be a key initiating event and driver of the disease. But nonetheless, because of its role in other tumor types, it's very reasonable to hypothesize that by developing drugs against brachyury, you might be able to make these other tumor types more sensitive to radiation, more sensitive to chemotherapy, or perhaps less prone to recur or metastasize. So based upon that evidence and that incredibly strong case for brachyury as a therapeutic target, we've recognized that the need to develop brachyury therapies is just at this point undeniable. The problem is that brachyury belongs to a class of proteins that have historically been very difficult drug targets. They are called transcription factors. There are hundreds of transcription factors that exist within human cells. They play really important roles in a variety of diseases. Almost every single cancer at the end of the day involves a dysregulated transcription factor, but they have been completely inaccessible to current or existing therapeutic modalities. Thankfully, however, just in the last several years, there's been a convergence of new technologies and new therapeutic modalities that make transcription factors, at least in theory, potentially druggable targets. However, this is still considered very high risk, and so you don't really see companies investing in this yet. There's starting to be some work in academic labs, but the reality is because it's so high risk, researchers over the years have come to understand that most NIH funding mechanisms won't support this work just because it's too high risk and there isn't enough preliminary data. And so really this is a body of work that is only capable of being supported through philanthropy. Thankfully, we have been able to partner with a couple other foundations that share an interest in demonstrating the potential of some of these new technologies to develop drugs against transcription factors. And together with them have begun investing in a portfolio of projects that aim to advance several different therapeutic modalities in parallel. And the reason that that's important is each of these is very high risk. We don't know if they're going to work, and we want to maximize the chance of success in the shortest amount of time possible. And to do so, we have to take multiple shots on goal. We have to place multiple bets. So essentially, these bets fall into 4 different therapeutic modalities, both targeting the brachyury protein as well as targeting brachyury at the RNA level before it even turns into a protein. We have implemented and are investing in a comprehensive, multi-pronged brachyury drug discovery initiative. This spans the gamut from developing a set of enabling resources like various tests that researchers can use, preclinical models, antibodies, things of that nature. Basically, the tools that every research lab is going to need to develop brachyury drugs. We've also, as I mentioned, partnered with a couple other foundations to invest now over $3 million in projects to tackle the earliest stages of brachyury drug discovery, all of which are making better than expected progress, like really exceeding our wildest hopes and expectations. We've also set up a number of services, like I described, the drug screening program, but these are services that are available to any researcher or any company that wants to develop a brachyury drug, so that those same capabilities don't have to be replicated over and over again. Upcoming, we see a need and an opportunity to make investments, venture philanthropy investments in companies, and potentially, if needed, we're prepared to actually facilitate the formation of new companies around promising brachyury assets that emerge. However, this is really, in our view, a last resort. The much better option would be to get these drugs into a capable, talented, well-capitalized drug company that will be capable of prioritizing them and moving them forward into the clinic quickly. I want to highlight for a moment, I'm not going to talk about all the brachyury drug discovery projects that are ongoing, but I just want to highlight what we consider to be the centerpiece of this strategy, which is a project involving several institutions, which aims to essentially develop the starting point for a lot of different types of drugs and create a set of tools that can be put in the hands of a lot of different researchers, a lot of different companies. These tools, or sometimes they're called chemical probes, or also sometimes called lead compounds, they serve 2 purposes. Number one, they serve as prototypes for drug development. So this is what a company would need to essentially take forward a drug against brachyury. They also serve as valuable research tools to put in the hands of lots of different academic researchers who could use them to study brachyury as a target in other tumor types, and thereby perhaps strengthen the case that the market for a brachyury drug could be bigger than chordoma, which is going to unlock the kind of funding that's needed to ultimately be able to bring these drugs into the clinic. And this is a really unusual and innovative project, not only because of the technology and the approach that they're employing, and not only because they're taking an industrial scale, undertaking an industrial scale effort, which is really quite out of the ordinary compared to most academic projects. What's really unique about this is that it's being done completely out in the open. There's no secrets, no IP, no data hoarding. Everything that they generate is available to the research community in almost real time. And all of the compounds that are developed are going to be freely shared. And why that's important is, typically when this work is done, it's done in private, and these compounds won't see the light of day until someone's filed a patent or until there's a paper that's published, which could be years down the road. We don't have time to wait. We want these compounds in the hands of as many researchers and companies as possible, as quickly as possible. This project essentially is a way to quickly and out in the open take the field to the 50-yard line, and then let people compete, let companies and researchers compete from 50-yard line to goal. But we think that we can move a lot faster, and we can go further with fewer resources, at least at the early stages of drug discovery, by doing this out in the open. So this team is making incredible progress. We first funded them in 2018. By 2019, they had the first compounds that would actually bind to brachyury, which was an incredibly important step. That was kind of the toehold to enable further discovery of these tool compounds that I described. Where they are now is compounds that are at least 2 orders of magnitude more potent. So binding to the brachyury protein much more tightly and at lower concentrations. But there's still more work to be done to get to the point where these can be handed off to other investigators and companies. And so now we're very actively working to line up the additional funds needed to close the gap here. And if that can happen, then it's very likely that within the next 18 months or so, we can get to the point where these compounds can serve as a starting point for companies to take them forward. So what does this all mean for patients? It's great that there's research progress happening, but at the end of the day, what matters is, how is this going to benefit from, how is this going to benefit those who are dealing with chordoma today, or those who might need better treatments in the future? Well, at this moment, if you're facing a recurrence, if you're facing a new diagnosis, if you're facing challenges that stem from treatment or the disease itself, there's a wealth of resources and support available through the Chordoma Foundation, through the online community Chordoma Connections, through our peer guides, et cetera. You are not alone, and there is a lot that can be done now. The reality is there's certain things that still can't be dealt with, and progress that needs to be made. But I think it's important to understand that you're not alone, and there is information, guidance, wisdom that's available. Additionally, there are more personalized treatment options that are coming down the pike and that are on the horizon. I mentioned the Pittsburgh biomarker, the Toronto subtypes. There's others that are coming, and with continued research, it's highly likely that in the next 5 years, we're going to start to think of chordoma not as one disease, but rather as distinct subtypes or distinct buckets, each of which need their own unique or personalized treatment approaches. For the patients who have advanced or recurrent disease, there are 5 chordoma-specific trials currently open. There's several more that are upcoming, as I described. In addition to that, there's 14 clinical trials that our medical and scientific advisory board have identified as being relevant to chordoma based upon what we know about the biology of the disease. And if you can't get onto a clinical trial, a number of those drugs are available off-label. And if anyone has questions about this, again, please reach out, support@chordoma.org. We can help decipher this and help guide you to sites that have trials open, or oncologists who are aware of all of these options. Additionally, as I described, pending the availability of resources, there should be several more clinical trials that make the leap from the lab into the clinic in the next year or so. And then behind that, a growing pipeline of additional promising concepts. The brachyury and the immunotherapy drugs that I described, best case scenario, these are probably 3 to 4 years away at this point from clinical trials. But in the scheme of things, it's a lot closer than they were even just a couple years ago. We've known and been talking about brachyury for quite a while now, and the reality is that the probability or the likelihood of these drugs happening in the near term, was not really high until recently. But now there is very clear line of sight and a clear path to bringing these drugs to patients in the near term, and frankly, on the time scale that patients who are living with chordoma today could benefit from. And so the final thing that I think is really exciting is that really we're at the point now where every single person in the community can meaningfully influence that timeline, whether that's by donating tissue to the Chordoma Foundation Biobank, whether that's by participating in the natural history study, whether that's contributing financially, whether that's doing a fundraiser. Literally every dollar counts. Every hand pushing allows us to move faster and go further. And there is now a very credible opportunity to make an impact in the lives of those who are dealing with this disease today. So tremendous thanks and gratitude to everyone in this community who has and is continuing to blow wind in our sails and who's come together to make this progress possible. There's a lot of excitement now, but the reality is it pales in comparison to what's coming. So I feel more and more optimistic, certainly more optimistic now than I've ever felt in the last 15 years, and look forward to working with many of you to bring about that better future that that we all desire and envision. So with that, I will wrap up and invite my colleague Sarah Nick to join me for Q&A, and look forward to answering any questions.
Hey everybody, my name is Sarah Nick and I'm the head of engagement at the Chordoma Foundation. It's so nice to be with you all today. It's really wonderful. We see so many familiar names here in the audience, and likewise, if this is your first event with the Chordoma Foundation, we're so glad you found us and joined us today. So for the Q&A, we've had some really good questions come in already, but Josh and I can stay and answer as many as we get. So please keep them coming using the Q&A box below. Josh, this first question is a two-parter. So the first part is there anything known at the moment about the factors that can contribute to the risk of chordoma recurrence? And then the second related part is, for the study looking at epigenetic signatures, if it showed that different subtypes had different prognoses, was different prognosis in that case synonymous with likelihood of recurrence or measured in some other way?
Yep. Okay, on the first front, very clearly subtotal resection is associated with greater risk of recurrence. Secondly, there's some evidence to suggest that the form of radiation and the dose of radiation is associated with recurrence. So if you've not had a sufficiently high dose of radiation and you are in the group of patients for whom radiation is warranted, that could portend a higher risk of recurrence. Beyond that, at this point, aside from the two, the biomarkers that I showed earlier, there isn't a really definitive way of predicting risk of recurrence. There are a number of other biomarkers that have been reported in the literature. One is called, for example, Ki-67, et cetera. But if you look at the paper that came out of Pittsburgh, they actually included Ki-67. They looked at it, and what they found was that the markers that they identified in their paper were actually more predictive of recurrence or outcome than Ki-67. So I think at this point, it's good initial upfront treatment, surgery, and/or radiation. On the epigenetics question, I believe that what they were looking at, the outcome measure was overall survival. And I don't believe that they were teasing out time to recurrence or propensity to recur. One other thing that I should note, though, is that both of those studies were done, well, the Pittsburgh study was done exclusively on skull-based tumors. The Toronto study was done mostly on skull-based tumors. Those studies need to be validated in a larger cohort of patients. And so that work is getting underway now. We've actually just received proposals from both of those groups, which we're hoping to be able to fund to basically validate the same exact work in a much larger number of samples derived from a larger number of institutions, and also importantly derived from tumors of all anatomic locations.
Great. The next question is there any possibility of restarting research into familial chordoma, and why or why not?
It's very much ongoing. So this is a project that's been going on at the National Cancer Institute for over 25 years now. The work that came out of the familial chordoma study has provided some of the initial evidence to point us towards brachyury as being a key driver of the disease. But that project is ongoing, and the group that's been running the familial chordoma study has continued to generate insights. In fact, they just published a paper this year in which they were not only looking at familial chordoma, but also at non-familial chordoma. So additionally, as a part of the natural history study, there's so the group that's running the natural history study is actually different than the group that's running the familial chordoma study, but they are linked up, and coordinating efforts at the NCI. So yeah, we continue to learn more, and certainly if anyone has been affected by familial chordoma, if you have other members of the family who have chordoma, exceedingly important to know for two reasons. Number one, there's a lot to be learned from a scientific perspective. Number two, if you and another member of your family have chordoma, that means that any blood relatives that you share in common could also be at risk for chordoma. And at the National Cancer Institute, as a part of their study, they will enroll blood relatives, unaffected blood relatives as well, and they will, for free, give them full spine MRIs to see, in fact, if they have any occult or undiagnosed chordoma tumors. And in fact, that has happened in some cases. So by participating in the study, there have been patients who have identified that they have chordoma and didn't even know it. So really, really important and very much ongoing.
Has there been any research on whether there's a correlation between chordoma and the risk of also having connective tissue disease?
That is definitely a part of the natural history study. There's all sorts of associations that have been postulated, asked about, proposed, whether it be pregnancy, whether it be trauma or injury from a motor vehicle accident, you name it. There's a lot of things that have come up over the years, or that people have wondered. The problem is you can only make, you can't really draw conclusions or generalize from anecdotes, and so the only way to really know whether there is an association is to study a large number of patients. But that's definitely something that is being studied and asked about as a part of the natural history study, is what other diseases have people been diagnosed with, what other comorbidities do they have, et cetera. And we hope that there will be some insights that fall out of that. I will say, just while we're on the topic of related diseases, there is one related disease that we know of that shares a common biological basis, and that is tuberous sclerosis complex. So tuberous sclerosis manifests in a number of different ways. It's a familial tumor syndrome. There's tumors of all sorts of organs that emerge. There's also neurologic and cognitive effects and other effects. And rarely, individuals who have tuberous sclerosis complex will develop chordoma. And similarly, sometimes chordoma tumors, even in patients that don't have tuberous sclerosis complex, have alterations in the genes that are responsible for tuberous sclerosis. So some risk factor there, but that's very rare. And to my knowledge, it's really the only other disease for which there is a strong association.
Next question is, what can you tell us about any recent progress or upcoming research focused on pediatric chordoma?
Yeah, thanks for asking. So it's been a big focus for us over the last 4 or 5 years, and really it's cross-cutting across, through all of the priority areas that I talked about, whether that be understanding the biology of the disease, the natural history, repurposing drugs, immunotherapy, et cetera. The key question we need to answer first is pediatric chordoma biologically different or the same as adult chordoma? Or more realistically, are there certain biologic subtypes that are enriched in the pediatric population versus the adult population, et cetera? And that'll determine whether pediatric patients ought to be treated similarly or different, and whether all the research that's being done for chordoma writ large can be applied to pediatric chordoma. In order to answer those questions, there's a couple things that are needed, and that goes back to the kind of research enabling resources that I talked about earlier. Tumor tissue, cell lines, mouse models. None of those existed for pediatric chordoma up until a few years ago. And thanks to the generosity of several families of pediatric patients, we've invested a lot over the last several years to overcome that barrier. So developed a number of pediatric chordoma cell lines, PDX models, et cetera. It's been more challenging to acquire pediatric chordoma tumor tissue, and so that's a big upcoming priority. We have worked with several researchers to include pediatric chordoma tumor tissue in their studies, but it's insufficient. And so there's more work to be done. And that's very much a priority in the coming years to build out our collection or a cohort of pediatric chordoma tumor tissue in the biobank, so that tissue can be included in all of the different studies that we talked about today.
We have just one last question, and it's a great one to end on, I think. Josh, if you had to pick one thing you're most looking forward to in the research and what you expect for progress against chordoma in 2022, what would you say that is?
Well, it's hard to pick just one, but I think, what is most important and exciting to me ultimately is impacting the options that patients have available who are in need. And so to that end, there are one or two, maybe even 3 clinical trials that could get underway in 2022. And that would, in the very near term, present new treatment opportunities for individuals in need. Looking out slightly longer, I think, resources permitting, in the next 12 to 18 months, there's going to be extraordinary advances that we continue to make on the Brachiora drug discovery front. And that will take us to the point, if not very close to the point of, being able to make a handoff to companies and kind of get Brachiora drug discovery programs kind of on a trajectory to enter clinical trials in the next several years. So that has been a long-term goal, long-term vision for me personally and for the foundation. Obviously, it would be so impactful for chordoma patients, for patients with other tumor types, and precedent setting. So just seeing what's on the horizon there is something that makes me very excited to kind of jump into action every day.
Same, lots of reasons to be hopeful. Thank you everybody for such great questions today.
Well, I'll second that. Really appreciate everyone's interest and truly appreciate the support that has made all this work possible. I mean, really everything we talked about today is the result of the collective investment and effort of hundreds, if not thousands of people across the world. I think the momentum that has been built up as a result is truly encouraging. So very much looking forward to continuing that in the year ahead, and looking forward to being in touch with many of you in the meantime, and to the next one of these webinars in the coming months. Meanwhile, feel free to be in touch. Feel free to follow us on social media. I hope you enjoy the rest of the day and the rest of the weekend. Take care, everyone.